Hello, my name is Connie Liu. I'm an undergraduate at MIT, and today we're going to be doing a worked problem dealing with nucleic acids. So, if you can see, I've drawn a little segment of DNA on here, and just as a recap, this little black circle would be the phosphate group. The blue pentagon in the middle would be our sugar. And the yellow rings on the side would be our nitrogenous bases. And remember, those can either be two-ringed, like right here, or single-ringed. Okay, so we talked a good deal about nucleic acids as individual monomers in the past, but we didn't talk too much about the bonds that happen in order to make it the polymer of DNA. So here we're going to be focusing on the green right here, and those are known as phosphodiester bonds. So phosphodiester bonds are what hold DNA and all of its nucleic acids together, and they form between very specific parts of the DNA. So the carbons on the sugar of DNA are always labeled. This carbon is known as a 3' carbon, and this carbon that attaches to the phosphate is known as a 5' carbon. So just as a interesting trick, if you ever want to identify the 5' side of DNA, you just have to find the side that ends in a phosphate, and that's going to be your 5' side. Alternatively, if you want to identify the 3' side, that's just the end that ends in the sugar, because the 3' carbon is right down here. So that's just kind of a little fun fact. If you remember, DNA has to line up in an anti-parallel fashion, so the directionality of it is really important. So identifying 5' and 3' can become super important in some cases. But let's go back to the phosphodiester bond. So this bond occurs between this phosphate group and the 3' carbon on the sugar, as seen right here. And this is what holds the nucleic acids together. If you want to form this bond, just as in all of the other polymers, a dehydration reaction has to occur. And dehydration reaction means that a water has to be removed, and then the bond can form between these two species, or the phosphate and the 3' carbon. So a water comes out, that bond forms, and then you have your polymer. In this case, that would be DNA. If you, so sometimes there are enzymes in your body that are able to do that. So the important one in this case is called ligase. The ligase enzyme is responsible for stitching these nucleic acids together. Say you have a break in your DNA, if such as, say, at Okazaki fragments. So at Okazaki fragments, the DNA is really disjointed because the DNA replication has to double back every time in order to fill in the gaps. So because it is so disjointed, the ligase comes in and patches those cuts back together. So it puts the nucleic acids together and forms a single string of DNA. So that's the importance of making nucleic acids into the polymer we all know and love, DNA.